So in a second, we'll take some questions from the audience. But I first wanted to kind of zoom out a little bit, and this session is about targeted sequencing. Can you talk about what motivated each of you to go down the targeted route? Is the primary driver that it's more cost effective, that you get an answer quicker, or are there some other factors that are more important?
Maybe I can start it off. For me, it's mostly the curiosity about the technology. So it's very fascinating for me to just see how far we can push it and what we can do. And ONT gives us these possibilities by having the APIs open source and allowing us to basically play with the technology and see what we can do with it. So that's why I gravitated towards this.
The ability to do new things. Amazing.
So in routine diagnosis, we are used to analyze a panel of genes. So it's quite difficult to perform whole genome sequencing because you will detect variation, but you won't be able to know whether these variations are causal or not. If you analyze a smaller panel, the answer will be more easy to do. It's just to do.
So for me, because the clinician asks more questions, not just the species identification. If we use adaptive sampling, we can get more information.
Wonderful, thank you. Do we have a question from the audience? Please stand up. Okay, we've got one back here, and there'll be a microphone for you. Fantastic.
Hi, thanks to all the speakers, one of whom I know very well. Given the location where we are today, I'd like to ask, what's the limit from the technology that stops you doing even better than what you're doing now? And what would you like to ask Oxford Nanopore to focus on next so that what you're doing works even better?
So for the moment, the limit of the technology is regarding the homopolymer region. There are some sequencing artifacts in the homopolymer region. You can manage this kind of problem with the quality score of the sequencing bases, but I think the main limit is here.
So for me, if we can combine many samples and adaptive sampling in one flow cell, it's safe.
And for me, I think there's still this open question about how much enrichment we can get, and there's so many factors that play into this. And so I think what would be really interesting for me is having a very, very precise control over, for example, things like fragment lengths, and being able to basically iterate experiments and isolate these factors, and then see how that affects the performance.
Great. So you've given us some homework to do. Is there another question from the audience? We have one here in the front.
I wanted to ask about the balanced adaptive sampling enrichment. I mean, are you using your own technology or bioinformatic tools to do this?
So we are not doing the sequencing ourselves, but we have collaborators who perform the actual sequencing experiments that I showed today in this little flash talk. If that was the question.
The actual question was, how are you balancing the different, is it in one run? It's in one run, and you're balancing the different taxa?
Yes, exactly.
But how are you doing? What bioinformatic tools are you using to do that?
For the different taxa,
yeah.
We don't currently do any kind of identification of the taxa, but we just base it on the data that we have observed. So we just take the reads and we fuse them together in an assembly-like process, right? And then we just take that as a basis for making the decision, irrespective of what the actual species is at the moment.
Is along the lines of your question whether that data is going out to another tool to look at the coverage and then coming back? Are you using Minnow, something built into Minnow, or are you using?
No, so for this we're using our own software. But I know that also for balancing barcodes, there's another team at Nottingham working on that problem specifically as well.
Great. So thinking about targeting, adaptive sampling is one approach, but there's capture bait probes, there's PCR, and I thought it was interesting that all of you are using adaptive sampling. And can you maybe talk about why that's the thing that you're doing for your particular project versus these other methods? Is there an advantage? What was the reason that you went with adaptive sampling?
Okay, me. Okay, so as I show you that we can get more information, this is one thing, but sometimes we use PCR to amplify 16S full length. So there are pros and cons, I think. Using PCR for 16S, we can, it's cheaper, yeah. So for adaptive sampling, we have to use one flow cell, but it's faster to get the result. Yeah.
For me, is that we are able to sequence native DNA, and by analyzing native DNA, you can observe methylation, DNA modification, and you avoid the introduction of PCR bias, and I think it's the best way to really analyze DNA.
Yeah, for me, it's also important to have the native DNA. Depending on the specific applications that our method will be applied to, I think in the metagenomics context, for example, it's very important to preserve these abundances as they occur in nature and then not introduce biases by performing amplifications. And the same goes for applications when we think about sequencing structural variants, things like that. And I think the actual coverage depth information that we get from raw reads is really critical.
Fantastic, thank you. Is there another question from the audience?
Hello, my name is Belen from the University of Murcia. I would like to talk about the adaptive sampling for the RNA. Have you ever tried adaptive sampling? Because I think that it's a bit difficult, this approach, but I think it will be very interesting to get a step forward to this approach to get higher depth in transcripts of interest. Thank you.
Never try on RNA.
I never tried adaptive sampling on RNA.
I also haven't tried it, but I would be very curious. And specifically coming back to a point I mentioned before, is the difference that you get in read lengths between RNA and DNA. And then there's again the question of how much enrichment you can actually get with the shorter reads that you usually observe with RNA. But I would be interested to see how it actually works.
Just to add to that, I think there are some folks in the audience, or certainly in the building today, who have tried direct RNA. So there's some early experiments with that. And I can connect you to have some conversations. And also, Hanli Ji's talk this morning was on single-cell RNA with adaptive sampling, although they're sequencing the cDNA from Tenax Genomics. So that may be of interest as well. Can you guys talk a little bit about looking a year or two into the future for your applications? What do you envision kind of that growing into or becoming for your projects?
So I think regarding the germline DNA and concept distribution, I think nanopore sequencing will be a good tool for structural variation next year or in 2 years. And I think in 5 years, it will begin, will be the, I think, a reference technology to detect germline alteration.
Fantastic.
Maybe one of the more immediate things that I think we're going to see is adaptive sampling on the Promethean, and then adapting our methods to that kind of data volume will be challenging, but also quite interesting, and I'm looking forward to seeing what we can do with that as well. But our software needs quite a lot of optimization to be able to handle that.
Yeah, so for me, my team tried to use adaptive sampling with many kinds of samples, not only the microbial kind of thing, but also cancer that my boss will present tomorrow. So, and now I start talking with many people to, if you have pus or abscess, something like that, please tell me. Yeah. and also the, not only human, in animal also. Yeah.
Thank you. Another question from the floor here.
I have used adaptive sampling on Promethean, and I was wondering, would you the software that you wrote, would it also work on Promethean, or is it only on Menayon for the moment?
Sorry, I can't understand
We couldn't hear
you
the
at all.
question. Sorry, it's real hard to hear from up here. Could you just repeat?
I will repeat. The tool that you wrote, does it also work to perform adaptive sampling on Promethean?
Oh.
Adaptive sampling on Promethean was the question.
Yes,
If
with your software.
I see. So you're running on a Minion, and I think the question is if the same pipeline you're using for balancing works on a Promethean.
We haven't tried that yet, so it doesn't work right now, and I think it wouldn't work out of the box, but I think that we can adapt it to work also on Promethean flow cells. But yeah, currently we're working with the Minion flow cells and mostly on the grid ion itself. And we're actually using the computational resources that are built into the grid ion to make all of these real-time computations.
Thank you. We have time for a couple more questions. Is there another question from the floor here? Another question in the back?
Regarding the adaptive sampling again, do you see differences if you are targeting one continuous bed coordinates, or if you split these megabases into different targets, like a panel of genes or something? The yield of enrichment, is it the same, or is it loose? I mean, is it not better if you do it in a panel rather than in a continuous coordinates?
That's an interesting question. Just about the amount, if I understand right, the amount of enrichment that you get, whether or not the enriched region is one contiguous region or you have a lot of genes spread across the genome.
We have a mean enrichment between 7 and 8 in a gene, because we analyze a complete gene from the promoter, the 3' UTR. And when you look at the coverage, you have a quite even coverage, but sometimes in some regions you could have a slight decrease or slight increase, but it's more even than with short read. In hybrid capture library, sometimes you have a big coverage in a certain part, and in another way, in another place, you have a decreased coverage. So with adaptive sampling, the coverage is quite even. Sometimes you have quite differences between different places, but it's quite good. It's quite even through the gene.
Any other comments on that? Okay, in the last couple seconds we have, what do you think the future is for targeted sequencing and adaptive? As sequencing costs come down, is it just going to be go back to sequencing everything, you don't need to target? Or as sequencing becomes more ubiquitous, is targeting going to be really crucial to deploying these kind of strategies? What do you guys think?
It's a good question. I think it's still going to be more efficient to use any kind of adaptive sampling strategies. You know, depending on the actual question, there's for sure merit in sequencing exactly what you want instead of just sequencing whatever's in the sample, in my opinion.
Yeah, I also think to answer this question is based on the questions what we ask. Yeah, and then put adaptive sampling or PCR or Cas9 or something. Yeah, depends on the questions. Yeah.
And for routine diagnosis, I think we will use targeting sequencing for a long time.
Wonderful, thank you. I'd like to thank all of our speakers today.